Hello everyone, hope you all are doing well. So today we are going to discuss the next copy of our Expire for Agriculture test series. So you all know that we had Expire for Agriculture test series in which we had 14 tests and we have discussed till test 9 and test 9 to test 14 were of full tests. So today we are going to discuss the test 10. So it is the full length test comprising of paper 2. Okay, so we will see whatever the questions asked and how the student has mentioned the answers, okay, or written the answers. So by analyzing this type of answers, you can get to know various things. You can learn from the mistakes of students. You can learn how to introduce, how to conclude, how to structure your answers. So all these things can be very helpful in your answer writing journey. And as I will be just discussing the structure important points. So if you want to read the answers line by line, you can simply pause the video and read it. I will be not reading everything line by line as it will be taking lot of time of us. So I will be just discussing the important points and briefly my uh, focus will be on structure of the answers, because all these things have been discussed by me in various answer writing practice videos, various static videos, so you can refer them for conceptual clarity. But you should more analyze here how students are writing. This is a copy of a good student, so you will be more beneficial in analyzing structure and pattern of the answers, how students are dealing with the answers. So that is the aim of this series. So the first question is, describe various factors affecting enzymatic activity in plants. So we all know what enzymes are. So enzymes are specialized proteins which help in speeding up the chemical reactions in plants. So these are what enzymes in. They are also known as biocatalysts. So this is how a student has introduced. Now, what are the factors affecting enzyme activity in plants? So first factor, obviously, high temperature. If the temperature is high, it can denaturate the enzymes. If the temperature is low, there could be if the temperature is low, there could be inactiveness in enzymes. pH is a factor. Enzyme concentration is a factor. Okay, substrate concentration is a factor. Salt concentration is a factor. So very well written points. You can simply pause and read the answers if you all want to. Chemicals are factors. Cofactors act as factors because they are essential protein structure of the enzymes. So very well written answer. Now let's move to the next question. The next question is, what is polyploidy? And give a brief account of classification of polyploids. So polyploidy is a condition in which plant have more than two sets of chromosome. Polyploidy. Okay. Homoploidy. You all know homoploids. Sorry, haploids. Here we have one set. Then we have diploids. Here we have two sets. Then we have polyploids. That is, here more than two sets of chromosomes can be found. So well defined in the introduction. Classification have been mentioned, because in the question only the polyploidy and its classification have been asked. So in classification, the student has mentioned about autopolyploids, allopolyploids, and examples have been also mentioned such as for autotriploid and autotetraploid. So very well written answer. And with these examples, whenever you add examples to your answers, it enhances your answers. So please try to write the answers. So please try to write the examples whenever you can. The next question is, describe the mode of origin and growth of cell wall and its functions. So we all know what cell wall is. Cell wall acts as a barrier against any kind of invasion of external agents. And we all know cell wall is generally present in only plants. It is not present in human bodies. Now, how cell wall is originated? Obviously, it is originated during the telophase stage. There is some deposition of pectins there. Then there is increase in pectin droplets. Then a union of various kind of plates is there. So all these things lead to formation of cell wall. And you can see how nicely the student has made drawing. Okay, at this stage, from this stage, you can see some cell wall formations, and at the last, you will find that the cell wall is fully formed around the cells. So very well drawn. Now growth have been also mentioned. There are two theories such as apposition and interception theory. So both theories have been mentioned, how apposition theory works, how interception theory works. So I think very good. And here I think function of cell walls can be avoided. Let's see it is asked in the question or not. Okay, yes, functions is asked, so please do not avoid it, you will lose marks. So you have to write some functions. So two three functions could have been written, and I think this one has written too many functions, and I think good, only if you have time you can write, but at least write 3 4, that will be enough. So very well written answer. Now if you want to read the functions, you can pause and read it here. You all know it: structure and support, protection, selective barrier, osmotic pressure. So these are the functions. Now the next question is, what do you understand by the vertical and horizontal disease resistance? So obviously resistant is disease, resistant from disease is very important for the plant to survive. Okay, the vertical resistance is, okay, here we have allogenic gene action, here we have polygenic gene action. Here in vertical resistance is the inherent ability of the crop to resist specific disease or pest. Here they can resist multiple diseases or pests simultaneously. So specific and multiple. This is the difference. Here it is oligenic, that is C12 genes, and it is polygenic. That is why it is able to, as there are various genes, it is able to resist from multiple diseases. So these are the differences. You can pause and read it if you want to. And again, well concluded. Now let's move to the next question. The next question is, explain the molecular marker approach in crop improvement. So what molecular markers are? So these are DNA fragments that can be identified within a genome and are used to identify the position of gene or a character inheritance. So well written and defined, I think very fair definition. And how it works, obviously there are identity markers, they work on selective traits, transmission is there, they create genetic diversity, reduce inbreeding time. So all these things are important, and there are various molecular markers which have been explained, such as RAPD, A, AFLP, RFLP. So very well written answer. SNP, morphological markers are also there. So this also shows how good students' knowledge base is. Or students has read all the important things, detail prepared well. That is why it reflects on your copy. Okay, so that is why basic preparation is very important, and practice answers that will enhance time management and all these things. You will through practice. Answer writing is art. However, knowledge base is important, but with practice you will develop your art. Okay. That is why reading and answer writing both are very important for UPSC, especially in maths. The next question is, describe the detailed structure of DNA as prepared by Watson and Crick with a suitable diagram. So you all know Watson and Crick prepared the double helix model. Okay, so that has been written in the introduction part. The structure have been mentioned, such as double helix, nucleotide, polymer, all these things have been mentioned. There are two purines, pyrimidines, that is purine, adenine and guanine, and pyrimidine, cytosine and thymine. All these things have been mentioned, and you can see a diagram have been made depicting all the things, that there is a sugar phosphate backbone, there are nitrogenous base pairs, okay, they are held together by hydrogen bonds. So all these things have been mentioned well. So good. Now silent features of Watson Crick structure have been also mentioned. That is right-handed double helix. We have strands in opposite directions. So all these things have been mentioned. Sugar phosphate backbone is there. So you can simply pause and read it. There is a connection through hydrogen bonds. A is generally with T and G is generally bonded with C. So all these things have been mentioned. So well written answer. Now let's move to the next question. The next question is, what do you mean by mutation, and what is the significance of induced mutation in plant breeding? So we all know what mutation is. Mutation is a sudden inheritable changes in genetic structure of DNA sequence of an organism. It may be acquired by some natural material or some artificial mutagens. So you all know it. Now mutation theory have been mentioned, that it is given by Hugo de Vries. So this is about theory. Now there are various types of mutations, such as point mutation, chromosomal mutation. We have other types such as all these mutations, silent mutation, and there is one more type, that is induced mutation. That is here it occurs in organism when it is exposed to mutagens in some labs, or induced. That is, it is not natural. We are inducing it artificially. So its significance have been mentioned. It is obviously in crop varieties improvement. Some examples such as DT10, it is a mutant rice variety, have been mentioned. Okay, about Rosen, it is a variety, have been mentioned. Cost effectiveness, quick, environmental friendly. So all the points have been very well mentioned. You can simply pause and read it. And what I have written here so that you can understand more emphasis to those parts should be given what is this means. If you look at the question is, what do you mean by mutation and what is the significance of induced mutation in plant breeding? So I think there was no need of this mutation theory. It could have been avoided, and that is what I mentioned here. Not all in the question can be skipped or written in brief. So that is what you have to keep in mind. And these parts, which is induced mutation, can be given more weightage because that is directly asked in questions. That will fetch you marks. I hope you understand this. Now, the next question is, discuss the role of public and private sector organization in seed production and marketing in India. So we all know what Indian seed industry's growth rates have been mentioned, that is 12 to 13%. So you can write about Indian seed industry in introduction part, that is fair enough. Now some organizations of involved have been mentioned. It is more both private and government organizations have been mentioned. So National Seed Corporation, state farms corporations, state seeds corporation, kvk, so all these are government organizations, and then some private organizations such as jk agri genetics, malayalam seeds limited, advanta seeds limited, pan seeds. So these are private organizations. Now here role have been mentioned, that produces self watered crop. Okay, all these things role you all know. Okay. Marks breeder seeds, focuses on public interest, enhances production, all these things. Both public and private sectors roles have been mentioned. So but this should be a bit clear, but okay, you have written, you will get marks on this. The fairly written answer, and I hope you understand all the things. And if you want to read the answers, you can simply pause the video wherever you want to and read it. So this was section A of test 10, and in the next video we will discuss the section B of test 10 only. Okay, so I hope you understand all the things. You will have like these questions, like this copy viewing. So I think that is helpful to you. So if you like it, please press the like button, please do share, and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis, and if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.